Okay, thank you. So let's start this afternoon session. The first speaker is here, Maria Bernabeu. She's from Spain. She studied, she made her doctorate with Hernando del Portillo in Barcelona. Then she moved for quite a long time to Seattle and is now back in Barcelona at EMBL. So she will talk about parasite surface interactions. Okay? Where's the mic?
Thank you very much, Daniel and Carol, for the invitation. It's very good to be here with this vibrant community. I did my PhD on vivax with Hernando del Portillo and spent some time in Manaus, so I'm happy to be here today. So let's go back. Okay, so my lab is working with bioengineering. So we are generating 3D microvascular models to study how malaria parasites break and disrupt blood vessels. So we're focused on the most pathogenic form of malaria, cerebral malaria, which causes, as you know, more than 500,000 deaths every year. And this is one of the main hallmarks of cerebral malaria, which is the accumulation of Plasmodium falciparum in the microvasculature, damaging blood vessels. You have here a normal blood vessel. And then eventually, this causes massive brain swelling in these children. So on the left, you have an MRI of a children that had uncomplicated malaria. And then on the right, you have children with severe malaria. And indeed, these kids die because they have this massive brain swelling that compresses the brainstem here, where you have the respiratory center. And we think that this is a consequence of blood-brain barrier disruption as a consequence of the parasite accumulation. But this has been very hard to study in the lab, mostly because we don't have good rodent malaria models to study cytodhesion. Cytodhesion in falciparum is caused by PFMP1, which is probably one of the most well-known variant families in Plasmodium, and it sits here on the knob surface. It's where you have the PFMP1. So it's so important for the parasite that the parasite has developed this machinery to export PFMP1 on the surface. It's also very complex. Each parasite strain has 60 different VAR genes that encode for PFMP1, and the parasite has this antigenic variation to avoid the immune recognition. And besides that, each strain has a complete different repertoire of PFMP1, so it's highly variable, highly difficult to study. But we know a few things about PFMP1. We know that it mediates this cytodhesion to the microvessels, mostly through these two receptors, EPCR and ICAM1. It can also bind to CD36, but CD36 has been associated to uncomplicated malaria, and EPCR and ICAM1 to cerebral or severe malaria. There's also a different variant that I'm not going to talk today, which is BAR2CSA, which binds to the placenta, causing placental malaria. And as I've mentioned, this cannot be studied in rodent malaria animals because Plasmodium berghei does not have PFMP1, so it does not have sequestration in the brain. So that's why when I was in Seattle, we started building these bioengineered models when I was in Joe Smith's lab. And this seems very complex, but mostly you have here two collagen layers, and one is flat and the other one has microfluidic pattern. And then we can seed primary brain endothelial cells in these 3D structures, as you can see here, and we can generate these tubes with a very, very complex microfluidic network. And we're using that because by using this kind of very geometrical network, we can recapitulate multiple different flow conditions in a single device, so we can test multiple blood flows in the same device. Okay, and then we can use it to perfuse malaria parasites. You've seen we have a tube, and this is a typical experiment that we do in the lab. We have the endothelial cells here forming this tube, and you will see that very quickly on we can perfuse these Plasmodium falciparum infected red blood cells, and just after 10 minutes we have massive accumulation of Plasmodium falciparum in our devices, recapitulating very quickly what happens in the brain of these children with cerebral malaria. And then I'm going to show you an example on how we can use these very complex microfluidics to study how immunity to malaria develops. It's very well known that cerebral malaria mostly occurs in children in sub-Saharan Africa, because as children get more malaria infections, adults become immune to the disease, not to the parasite. So this immunity is building, and we thought that this was through PFMP1. And then what we did in this study in collaboration with Thomas Lapsten and Evelyn Bannick is to understand whether adults in Africa and in endemic countries can have these antibodies that prevent this cytodhesion. And as I've mentioned, there's 3 different receptors. So in this study, we focus on this one, EPCR, because during my postdoc, we also did several studies, both in India and in Africa, showing that cerebral malaria patients always have these parasites that bind to this receptor that is very important for the health of blood vessels. So we talked this morning, this parasite is highly variable, as mentioned already, and these PFMP1 variants that bind to EPCR come in 6 different flavors. And what Thomas found, he has this amazing library of proteins of 7 different strains targeting most of the variability of PFMP1. And we isolated antibodies, Evelyn, from Ugandan adults. And we tested these antibodies that you can see here with these recombinant proteins. And we found that there were 3 of them that we were very excited about because they bind and inhibit binding to EPCR in 5 out of these 6 different variants, which was very exciting. Then Thomas also did a lot of cryo-EM studies, studying how the protein binds with the antibody. And what he found is that the protein and the antibody is completely blocking this interaction site between EPCR and PFMP1. So this is where we come in. Thomas reached out to us, say like, okay, I have this protein that I think is very inhibitory, but we cannot test it in animals. We can do very simple in vitro models, but what if you do yours? And then we did this, so you can see here in this graph, here at the bottom, we have the shear stress, and what we see is that the parasite binds with this shear stress-depending binding, so it has very high flow at low shear stress, and it binds slower when the flow is faster. And what we found is that this protein monoclonal antibody, let me go back, inhibit parasite binding at all shear stresses. We tested this with also different strains. This was very difficult to get because we cannot work with 3D7. 3D7 also does not bind in our system. So we had to really clone a new parasite line to test it, that these antibodies can really inhibit parasite binding. Okay, so changing gears, one of the main focus of the lab right now in Barcelona is to study how Plasmodium falciparum breaks the blood vessels and causes blood-brain barrier disruption. And for this, I've shown you our model. It only had endothelial cells. But to have a good blood-brain barrier, we needed other cell types. And the blood-brain barrier is the structure that you have in your blood vessels that has very selective transport of molecules from your blood to the brain to prevent brain damage. And of course, endothelial cells are very important, but these 2 other cell types, astrocytes and pericytes, play a key role in providing these barrier selective properties. So we have these two cells that surround the endothelial cells, and through a crosstalk, they make this barrier very selective, so the junctions in between endothelial cells in the brain are very tight. And also we have a lot of transporters here that are causing selective transport of molecules. So what we did when I started my lab in Barcelona was to introduce these two cell types in the model, astrocytes and pericytes, and then we put them in the collagen hydrogel. And then when you put all these cells together in culture, magic happens, and the cells know what to do when they're in culture and where to go. So after, say, 7 days in culture, what I'm showing here is the endothelial cells are staining white. And then you can see here a pericyte in pink and an astrocyte in green. So they migrate towards the blood vessels and establish these interactions. As you can see here, for example, astrocytes have this end feed connecting to blood vessels. And we can have these beautiful 3D reconstructions showing a very similar morphology here than in vitro. And more important, when we put these three cell types together, we have a much better barrier in our microvessel models. So this is a permeability assay in which we put a fluorescent tracer with very, very low molecular weight. And you can see that when we add these three cell types together here, our vessels become much, much more impermeable. And this is perfect for us because then we can do our studies with Plasmodium falciparum. So then what we did in this study is to take different synchronized stages of the malaria parasite, very tightly synchronized, to look which one was more pathogenic. And for this, we did a lot of single cell. And today, for the sake of time, I'm just going to focus on this condition that we call egress. So it's when the parasite ruptures and releases all these products into the circulation. And we did a lot of single cell here. Alina did that. And it was very exciting to see that when we add these egress products from the malaria parasite that you can see here in orange, our endothelial cells completely change the transcriptome, not only endothelial cells, but also pericytes and astrocytes, which are the cells that are across the barrier, showing that these malaria products were also reaching beyond the endothelial cells. And then here, I don't think you can see, but what we found is that in these three cell types, we had the same pathways being upregulated. This was mostly interferon type 1 response. And one thing that really caught our attention is this JAK-STAT pathway that I'm going to talk more about it later. And this was massive. So when we add these egress products, here you have a normal vessel where you can barely see an expression of STAT1 in green. And when we added the egress products, it became like super green, showing like a high activation of JAK-STAT. Then we also look at downregulated pathways, which were also very interesting for us, because we found that there were less genes being downregulated, but these were very important because we found that these genes that are responsible to keep the junctions between the endothelial cells were all downregulated in this single cell RNA-seq. And you can see here, this is an immunofluorescence. This is a control device perfused with red blood cells. You can see there's like beautiful junctions. They are very, very thick. And when we have the malaria, these junctions become much thinner. So we don't have thick junctions, and we started seeing like the appearance of these big gaps and these big holes in the blood-brain barrier. Then, as I've mentioned, we also did a lot of permeability assays. In these assays, as I've said, we put this fluorescent low molecular weight tracer, and we see how it leaks to the collagen. And we barely have any leaks here when we have the control, so like the dye stays inside. But when we add egress products, there was leakage of the blood products into the brain side of our engineered models. So then we wanted to connect these two different findings, the decrease of junctional proteins and increase of JAK-STAT. And for this, we took advantage that JAK-STAT is a pathway that is dysregulated in many inflammatory diseases. And there's many drugs that are on the market already available to then regulate this pathway. And for this, what we did is we treat our devices with ruxolitinib, which is this JAK-STAT inhibitor. And we were very excited to see that by using this anti-inflammatory drug, we could prevent this barrier breakdown in our 3D microvessels. So we found that the barrier properties were even better than the control when we add this drug. When we look at the junctions, I don't know if you can see down there so well, but there's barely any junctions here, tight junctions. And when we add this ruxolitinib here, we could recover these very nice junctions in our 3D model, showing that an anti-inflammatory drug can potentially reverse this blood-brain barrier breakdown. And this is very exciting because there's been some CHMI, some controlled human malaria infections done with ruxolitinib, mostly led by Michelle Boyle and Christian Nguerda in Australia, and they have used it for uncomplicated malaria. So they've shown that when they treat these uncomplicated patients with ruxolitinib, this drug attenuates the inflammation. It does not prevent the acquisition of immunity. And in this paper, they show that there was an improvement of vascular dysregulated pathways. And now it's very exciting that with our model, we can test how ruxolitinib can also preserve vascular integrity in our 3D model. Okay, and then I would like just to finish with probably one of our longest projects in the lab and the most fundamental. And in this project, what we did is we focused on the interface between endothelial cells and malaria parasites, and we did in 3D using volume electron microscopy. When we did the single cell with the trophozoites, for example, we could barely see any transcriptional differences, but you're going to see how there's a lot of differences and changes that happen at the cellular level. And Hannah is doing these beautiful 3D electron microscopy studies that you can see here. So you can see two endothelial cells holding together. We get this in 3D, infected red blood cell on top and the parasite, and we can really have like very high resolution of how these two endothelial cells interact and this interaction with the parasite. We could look at the junctions, but we didn't see, there were some differences, but not too much. And then this was a very hard study that Silvia knows Hannah very well. It took a long time because at the beginning we were trying to do this EM and we could not find these regions with parasites. We were finding only endothelial cells. So then we had to bring our devices, fix them in Barcelona, treat them for EM, and bring them to Hamburg. Hamburg is in the north of Germany, and we will run these samples on the synchrotron in an X-ray modality, so it's going to the doctor, but in a big machine. And then we had super good resolution of our devices, and you can see clearly the endothelial cells and the infected red blood cells here. They can see like very nicely how they interact with the blood vessels. And then with this, Hannah brought these devices to Heidelberg, which is in the south of Germany, to do this volume electron microscopy. It's known as FIB-SEM. Basically uses a laser to cut down the sample and image. And then with this, we get these amazing, beautiful images. So this is a slice, but then we can have a 3D reconstruction of how the malaria parasites are interacting with the endothelial cells. And one of the things that was most striking is the appearance of these cap-like structures in the endothelial cells that are causing these projections in the interaction site with the infected red blood cell. So the endothelial cells are hugging and wrapping up the parasite. And then we also look at the knobs. So I've mentioned at the beginning of my presentation, knobs are these structures. You can see them here. This is also a 3D structure of an unperfused parasite. And this is a very cross-linked structure. So you have the spectrin cytoskeleton in which the parasite is putting proteins and creating these structures that anchor the PFMP1 here at the surface. And this was thought to be like a very rigid structure, but we have found that this is not the case in our 3D model. Before, the schizen's were described like as this big ball, like a tennis ball when the parasite is inside. But we found that no, that there's a big remodeling of the infected red blood cells. So there's like these folds and groups in this interaction site with the infected with the endothelial cell. And with this, we turn to biophysics that sit next to our lab in EMBL Barcelona. And what they did is a very careful segmentation of the knobs. So they can identify very nice where the knobs are in this infected red blood cell. And what really, really surprised us is that these knobs were not evenly distributed on the surface. They were much more enriched in these regions where the folds are. Here you can see in this orange. And then on the side of the infected red blood cell that is on the lumen, they are very lowly present, and they concentrate mostly in this interaction site. And then because they are mathematicians and they can do super cool stuff, what now they are trying to do is to see the forces that the infected red blood cell is making on the endothelial cell. And you can see in these arrows here that in these areas where the knobs are enriched, the parasite is really, really pushing towards the endothelial cell, which we think that is also causing this remodeling of the endothelial cell. So now we're looking at what comes first, whether it's the endothelial cell causing these changes and then the infected red blood cell like answering to that and responding to that, or whether it's the parasite first initiating these changes. But more important is like, does this have any consequences? And for this, what we did is to inhibit the formation of these structures here with a drug that inhibits actin in the endothelial cells, and did one of our complex flow experiments in our system. And what we found is that when we inhibit the formation of these structures with this drug, it's known as CK666. It's an actin inhibitor. What we found is that when we added flow after the site adhesion, we could flow away the parasites much more easily. And then when we had like normal conditions, this parasite remains attached. So we showed that this formation of these structures is key for the interaction between the infected red blood cell and the endothelial cell. And with this, I would like to finish. My time is over. But very quickly, I really want to give you 3 examples on how we can use our bioengineered systems to study important questions in malaria research. We first showed that we can study how immunity to disease develops in cerebral malaria and how these monoclonal antibodies inhibit parasite binding. We can also show that malaria parasites falciparum causes blood-brain barrier disruption when they egress, and this is because of a combination of JAK-STAT inflammatory pathways and downregulation of junctions. We could reverse this with a JAK-STAT inhibitor, which is very exciting for a future potential treatment for cerebral malaria. And finally, we had this beautiful study on EM showing how these 2 cell types interact at extremely high resolution and how this increases the affinity of the parasite to endothelial cells. With this, I would like to finish. And this is my lab at EMBL Barcelona. So I would like to thank all the lab members. These are very extremely difficult experiments to do, to coordinate the engineering with the malaria parasites, and to all our collaborators. Thank you.
So the next speaker, thanks a lot, Maria, for this very nice pictures and nice talk. So the next speaker here is Leonardo. He's actually graduated in pharmacy and biochemistry in Juiz de Fora, Universidade Federal Juiz de Fora. Then went to Fiocruz in Rio, has a specialization at the National Research Center on Protozoan Diseases in Japan in '96, a long time ago. Then he did his doctorate at Fiocruz too, and now he's a pesquisador titular, I don't know how to translate that one, main researcher at Fiocruz. The floor is yours.
Obrigado, Gerhard. Em primeiro lugar, obrigado, Daniel, Carol, Beto.
Thank you, Gerhard. First of all, thank you, Daniel, Carol, Humberto, all of the organizers for the invitation to speak here. Very well organized, very nice event. Congratulations. I'm going to speak a little bit about the work that we have been doing in experimental malaria. I think Maria's presentation gave us a good outline of the breakage of the blood-brain barrier, which has to do with what we're going to speak here. It's not directly related, but it has to do with it. So I'll talk about the role of leaked plasma on vascular constriction in experimental cerebral malaria. Just to speak about the problem of cerebral malaria, it is the most lethal complication of malaria, of Plasmodium falciparum, and there's a treatment which is intravenous. It's an intravenous medication, but it causes 15 to 25% of treated patients to die. We have been working on an experimental model to try to understand how this process happens with intravenous artesunate to develop therapies that improve this outcome. As Maria showed very briefly, just the pathology, malaria is a hematological and vascular disease, and severe malaria is very severe to the vascular system. It is characterized, as Maria said, by the parasites in the endothelium of the brain that might lead to an inflammatory, very intense inflammatory process with a lot of leukocytes in the brain. There's a presence of microhemorrhages everywhere. This rupture of vessels, we have the breakage of the barrier, and there's the rupture of vessels, and that leads to a very serious hyperfusion problem. One of the characteristics of this vasculopathy in severe cerebral malaria is that you have a problem of endothelial dysfunction. There is a very harmed endothelial response, even in moderate malaria. There is a process of vasoconstriction. So with the blockage of vessels, the process of vasoconstriction leads to a decrease in the blood flow to the brain, a process of ischemia. And in this work using Doppler, it shows that there are 4 profiles of vascular alteration in patients with cerebral malaria. Patients with low flow have a higher lethality than other profiles. One of the biggest problems in vascular issues is that the patients with severe malaria or cerebral malaria have a low bioavailability of nitric oxide. It is a small molecule that is produced by the endothelium through the oxide synthase enzyme, which leads to the production of nitric oxide, which is taken to the smooth muscle, leading to the production of cGMP that is going to take to the dilation of the vessel. In malaria, you have a low bioavailability, and this whole process of dilation is harmed. As Maria said, we use an experimental model that does not reproduce all of the characteristics of the human malaria, cerebral malaria, but it reflects pretty much everything that happens in the cerebral malaria in humans, including adhesion, not as intense as it happens in cerebral malaria, and a very serious inflammatory process, a process of vasoconstriction that we demonstrated that reduces the blood flow to the brain. And you can see here that in a regular animal, you have an intense brain flow here. This is done by laser Sperkel. And when the animals get the disease, they have a reduction in flow that is progressive until it gets to a point that it becomes very intense, leading to the animal's death. And what we showed is that in addition to this process of vasoconstriction, vascular constriction that decreases the blood flow to the brain, the animals also have a harmed endothelial response. If you stimulate the vessels of this animal in vivo with acetylcholine and N-methyl D-aspartate, the animals respond dilating the vessels, and the animals in brown here, they do not respond. So it is constricted, but when you stimulate to dilate, they do not respond. This is in vivo. The interesting part is that when we studied that ex vivo, we were wondering if this non-dilation, this process of constriction, was due to an intrinsic defect of the vessel caused by factors of the parasite or inflammatory factors or anything that could be affecting during the process of the disease. And unexpectedly, what we observed is that ex vivo, the animals respond, both the ones in control and with cerebral malaria, they respond very similarly to the stimulation through the production of nitric oxide. And also the constriction of the vessels with serotonin is maintained normal. So what we observed in vivo was not reproduced in ex vivo. So we were wondering if the vessel, since it does not have this intrinsic defect caused by the contamination, that could be an environmental factor. The vessel that is in the brain, we think about the everything that surrounds it, or in the case of plasma, other factors of the parasite or inflammatory factors that could be affecting the function of the vessel. So you have this dichotomy. In vivo, you have a process of strong vasoconstriction with very little dilation, and in ex vivo, you have a normal behavior. The question that we asked was, is the vivo environment driving cerebrovascular dysfunction in ECM? And the basic hypothesis was initially to evaluate factors in the plasma, if there's anything circulating produced by the parasite or coagulation that could be affecting the stimulation of vasoconstrictor effects. This is to show you how the experiment is carried out. We infect the animals. We wait until day 6 because they develop a neurological syndrome. They develop seizures, ataxia. They are comatose or have other characteristics. So we evaluate parasitemia, temperature, clinical signs to assess the stage of evolution of the disease in animals. And we did 2 very simple procedures. An ex vivo study where we collected the arteries, the cerebral arteries of the animals, the plasma from control group and the sick group, and we assessed the ex vivo vascular function. And an in vivo study, if I have time, I'm going to show you some preliminary outcomes for the evaluation of the cerebral blood flow in these animals. For the ex vivo experiment, we used a procedure called mygraphy, wire mygraphy. We use an equipment where we have a chamber where you place the vessel, you put a wire through it, and it causes a tension on the vessel, and you can assess how the animal responds to stimuli. For example, stimuli we put in the fringe, which is a vasoconstrictor, is going to increase the pressure. If you use acetylcholine, which is a dilator, it comes back to the regular tension. So we started doing this with different concentrations of control plasma and infected plasma. And what we observed with a small concentration is that the plasma, starting with 1% and 3%, started to contract the vessels. So apparently the plasma has a constricting factor. And the interesting part was understanding that the control plasma and the ECM plasma had the same effect. So it did not seem like a typical characteristic of the sick animal. And as I showed before, nitric oxide is the main physiological dilator, and we wondered if there would be a problem in the production of the nitric oxide in the system. What is happening is that if you stimulate with plasma purely, you have an increase in tension, a constriction, but if you eliminate the production of nitric oxide with this inhibitor, the constriction increases a lot in the control plasma and the infected animal's plasma. So apparently what is happening is that the plasma has a constricting factor, and this constriction is strong, but physiologically it's counterbalanced by the production of nitric oxide. If you block this production, you increase the constricting factor. So we did not see a difference between plasma from the control group and the infected group. Just to reinforce the profile of the experiment, we stimulate to see the functionality of the vessel initially, and then you add the inhibitor of nitric oxide in the beginning, and then you stimulate with the plasma. And from 1 and 3%, especially when you inhibit the production of nitric oxide, you have a very strong constriction. So what we are observing is that if you add plasma, it induces constriction, and the vessel is going to produce nitric oxide to counterbalance. And just to look at the mechanism, we saw that this plasma-induced constriction was dependent on calcium. If you add a calcium chelant, you inhibit completely this process of constriction. And then we asked this question. Now, those who conducted all of these experiments was a doctorate student, Guilherme Sanches, and he has been conducting all these experiments. And when we got to this point, we knew that there was a factor in the plasma, and we discussed what could be the possibilities. Since the control plasma was also doing that, it seemed physiological. So he suggested serotonin, because serotonin is produced by the enterocytes and is captured by the platelets, and the platelets work as a storer of serotonin that is going to release it when it's necessary. And in cerebral malaria, it's proven that platelets have a very strong role in the pathogeny of cerebral malaria. And we were wondering, with the release of serotonin, that could be a factor in this process. And in order to do that, he proposed the use of an inhibitor of one of the producers of serotonin, HTL2A. And what he saw was that in the first evidence, if you do the same procedure of constriction using plasma, if you add ketanserin, you inhibit in 80% this constriction. So apparently, with the control plasma and with the ECM plasma, you have the same profile. And apparently, most of this constriction is being caused by serotonin. It does not exclude the participation of other factors, but it's a strong factor.
So these are the conclusions of the first part. The isolated vessels of cerebral malaria. They are preserved, and if the plasma are inducing the constrictions on isolated aortic rings, the constriction is counterbalanced by the nitric oxide, and the blame plasmatic factor inducing constriction ex vivo is the serotonin. So if it's coming from the platelets, how could it activate this smooth muscle? We try to see if, in fact, the stimulation of plasma, because the previous system was an open system. So the luminal and abluminal parts of the vase are exposed to the plasma and the inhibitors. And here we used a system that was very elegant, where he had a myography called pressure myography. And in reality, you put the vessel here, and you could perfuse the vessel in the lumen, or you could subperfuse from the external part. So he tested putting plasma or serotonin on the outside or only in the inside. So what he observed was that when you had the control plasma and the disease plasma, when they are inside the vessels in the luminal phase, there was practically no construction, but when you put that outside of the smooth muscles, you could see this constriction, and that was valid for plasma and for serotonin, a very similar profile in both cases. So this is conclusion number 2. Both plasma and serotonin, they elicit enhanced vasoconstriction via luminal stimulation compared to luminal exposure, and the stimulation with nitric oxide production counterbalances the serotonin-induced constriction. A mechanism proposal, since we are trying to advance, Guilherme is going to have his fellowship at NIH with the malaria research program. He's going to spend 3 years there working in a proposal. And one of the issues that are uniting the data to what Maria just presented to us is that if you have this hematocephalic barrier with neurovascular unit, you have this you have the arteriolar constriction, and you have actions that could disrupt the vessels, and then you have the proximity with arteriolars with the smooth muscle, and that could induce the constriction with the serotonin. So that means that in this cerebral malaria process where we can see this barrier being disrupted, the stimulation of plasma in the smooth muscle has no importance if you have a whole barrier. If it's broken, the plasma is going to escape, and parasite factors, inflammatory factors are going to produce several changes, and one of the possibilities is that it's going to take this to this constriction process that is going to prevent the supply of blood to the brain, and that would make the case more severe. Some preliminary data on in vivo studies. Guilherme was testing the blockage of serotonin with the receptors of smooth muscles, that if that would improve the blood flow with animals with cerebral malaria. So in fact, the animals, we inject ketanserin on days 4, 5, and 6, and then we measure the cerebral blood flow in the animal using this laser speckle methodology. And what we observed was that the ketanserin did not improve the blood flow at ECM onset. So before the infections like that, here the infected animals treated with ketanserin or the vehicle with no difference. On day 6, when they develop cerebral malaria, and 6 hours after the treatment, no difference at all. So ketanserin has some limitations. For instance, if you have higher concentrations, it induces hypotension. It's hard, and the animal is hypotense, and that will hamper the blood flow, and it has a low half-life. So we try to improve the blood pressure of these animals so that somehow that could help the ketanserin effect. And when we administer, that we recover the blood pressure in the animals, but it doesn't change the cerebral blood flow, even with a better pressure. So the increase of blood pressure is being counterbalanced by the adhesion or vessel blockage or the swelling. And this is preliminary data, but when we add ketanserin with norepinephrine, you're regulating the pressure, and then you're start with catastrophe, you have an improvement of cerebral flow in the animal. This is not meaningful. We didn't have many animals, but maybe we could have more interesting results. So the conclusion number 3 is that the ECM induces a progressive decrease in cerebral blood flow. Ketanserin does not prevent CBF reduction during ECM, according to this protocol and concentration, but the adding norepinephrine tends to attenuate the progressive decline in CBF during ECM. Most of this work was developed by Guilherme. The picture was taken in April 25th, 2022, 4 years before today. Guilherme was conducting most of the experiments with Fabiana Gomes, Melina Merloni in the malaria lab, with other collaborators, and especially at we had Cristiano Ponte with the Applied Science Center, and Thais and Fernando Senada from the Laboratory of Electrophysiology. And the pressure studies was made with Hans Ackermann and Abdul Hakim. And these are our funders. Thank you.
Thanks for the nice talk, Leonardo. I would like to call the last, oh, she's here. The last speaker, Sabrina Epiphanio, from the pharmacy at USP. She actually studied, she got formation in veterinary medicine, did her master and did her master at USP also. And then she did a doctorate too, yeah. And then she did a postdoc at Gulbenkian in Lisbon. Came back to Brazil, worked for a time at the Federal University of ABC near Sao Paulo in Diadema, and then came back to USP, where she's now associated professor at the pharmacy department. Okay, please.
Thank you, Gerhard. Boa tarde a todos e todas.
Good afternoon, everyone. It's a great pleasure to be here. I'd like to thank the organizing committee, especially Caroline and Danielle. Thanks for inviting me. It's an honor and a pleasure. To start, I'd like to talk a little bit about the model that we've been working with in the last 16 years. So it's an ARDS model, acute syndrome associated to malaria. And macroscopically, we have the animal presenting an increase of permeability and a red lung with a lot of congestion. And that could be also observed with this X-ray change. This is an infected animal with ARDS. So besides the inflammatory process, we have this diffuse edema with hemorrhage areas, and this swelling is one of the marks of respiratory disease and one of the causes that will take the animal to hypoxemia and death. And comparing with a non-infected here, and we also observed that these animals who develop ARDS, they also present a greater amount of infected erythrocytes in their lungs. And we also observed the quantities of those who were developing ARDS and not developing ARDS. And when we quantified the accumulated emulsin in the lung of these animals. So this model is an infection model in which we use the mice infected with Bergen-Anker plasmodium. And those who are developing ARDS, they die between day 7 and 12 of infection. So this model started 16 years ago when I was studying my postdoc at Bank. That was this first work when we described the model. And then as the main researcher, we better characterize the model to develop this predictive model to identify the animals dying from ARDS in on day number 7 of infections compared to those who didn't develop. And on this first part, I'm going to talk about the outcomes of these studies, the main research line of my lab. The first point I'd like to highlight is that here we may see in the mice mice with ARDS, this interaction between the endothelial cells and the infected red blood cells, showing that there's this cytoadhesion. And we also identified the ECAN and VCAN receptors. They are in greater expression in the animals developing ARDS than those who do not develop. And here we also observe EPCR because they were underexpressed in animals developing ARDS compared to those who do not develop concerning protein and genic expression. So in the assay that we had the knockdown of EPCR, we observed that from this knockdown, there was this smaller cytoadhesion of the endothelial primary cells of mice isolated from lungs compared the non-infected and non-stimulated because there was no knockdown. Since we have this increase of permeability and also the swelling of the lung is an important mark. We want to understand what are the mechanisms that are causing this increase of permeability. So from isolated cells from mice lungs, endothelial cells, and those are non-stimulated, and here we have the stimulated with infected red blood cells. And we observed this structure of the cytoskeleton of these cells and an increase of intercellular spaces. And we also observed this change in the microtubes when we stimulate with infected red blood cells. When we identify the microtubules, they have this smaller expression due to compared to the non-stimulated ones. So we also observed that in this assay of permeability increase, this is one of the assays that we developed from semi-permeable membranes. We stimulated these cells and then we put this dye called Evans blue, and then we measured the amount of Evans blues in the inferior compartment, and we saw that the infected red blood cells presented a greater permeability. So we had that compared to non-stimulated or the stimulated without the physical contact of the IRBCs with the endothelial cells and or with the non-infected RBCs. Another mechanism to increase permeability that we identified in our model was the cell death. So we observed endothelial cells marked as tunnel-positive, and when we had the assay again with pulmonary endothelial cells for mice, we observed that within 24 hours of infection, here we have 24 instead of 1 hour. The cells start to, to suffer with apoptosis, and also this apoptosis was made in this in vivo assay in lungs with different caspas. In this in vitro assay, again, with semi-permeable membranes, we observed that there is the same result, the increase of permeability with the non-infected cells. And when we used this, the pan-caspases inhibitor, we observed that this increase of permeability was not proceeding. So it inhibited that formation. So cell death is a different mechanism to increase permeability that will generate edema. So in vivo, we had our model using this caspases inhibitor, and we've shown that the animals survived for more time, and there was no difference in parasitemia, and the respiratory frequency of the control animals was improved than those who weren't treated. So this is the lung of a non-treated animal with hemorrhage and severe edema, and this animal to the right was treated, and both were infected. And another mechanism that we studied was the formation of the inhibition of NAT of gnats, improves malaria-associated ARDS. So they promote the formation of these gnats for in vivo and in vitro models, and that when we inhibit this formation, the neutrophils, using civilistat, which is an elastase inhibitor, or homozyme, which is a drug used, we were able to revert the ARDS case. So the animals survived more with homozyme and civilistat, and the lungs and their cytology remained less affected when compared to control animals here. So, in this first part, we show that the infected red blood cells are associated with ARDS development, that the VCAM and EPCR receptors contribute to ARDS pathogenesis, as we have with falciparum. So the proteins are different. They do not form knobs, but there's this contact. Using these receptors, we are able to modulate the pathogenesis,
and that the contact with the infected erythrocyte with the pulmonary endothelial cells is going to promote an alteration in the actin in the microtubes. It will induce the formation of nets, induces apoptosis, and it leads to increased vascular permeability. The blocking of these caspases and neutrophils prevents ARDS. So we were interested in knowing which are the proteins, the signature that ARDS develops, that it displays in animals that develop ARDS. We did then a proteomics of the lungs of the infected animals and the non-infected animals. We did a proteomic mapping. We identified 146 proteins, and out of them, 42 were exclusive to the animals that developed ARDS. In these proteins, we had the inflammation pathways, things that we had already validated, activation of toll-like receptors, and some proteins were identified as possible biomarkers. Among them, septin-9. When we saw this result, we wondered, what are the septins? What is the septin? They are considered the fourth element of the cytoskeleton. They are proteins bound to GTPs. They are involved in several situations of cell mobility, phagocytosis, endocytosis, adhesion, and cell migration and division. And when we continued observing, septin-9 specifically was bound to actin and tubulin, in addition to other adhesion to microfilaments and close to the membranes of the cells. And in this paper, we identified that the septins regulate the junctional integrity of endothelial monolayers, like V-E-cadherin. And when we did the knockdown of this septin here, septin 2, we had a decrease in the expression of V-cadherin. And looking at our results, we had a decrease of the expression of different proteins related to adhering junctions, occlusive junctions, and communicating junctions. That's the case here of connexin 43, occludin in adherence junctions, and V-cadherin and the GEM molecule. And another piece of information in our searches for who would be that septin-9, we saw this paper. It was very illustrative in the sense that the motility of the cytoskeleton can be carried out by the activation of Rho-A. Rho-A is going to activate the ROCKinases here that promote the phosphorylation of the light chain of myosin, and this phosphorylation leads to the to myosin. That corroborated the results that we had in relation to the activation of Rho-A. When we stimulate the primary endothelial cells of these mice with infected erythrocytes, we observed a greater activation of Rho-A and also a greater expression of the light chain of phosphorylated myosin, showing that there is this contraction. And just to show you here this adhesion molecule, an adhesion kinases, which we also observed later that it reduces its expression when we stimulate with infected erythrocytes, corroborating this article that we studied. So that made us pay more attention to the septin and wonder what would be the role of this septin-9 in a ARDS murine model. Because there is a contradiction in literature that in some models, superexpression increases, for example, permeability, ocular permeability, leading to an increase in glaucoma. Another model, when you do the knockdown of septin-9 in a model of permeability, intestinal permeability model, there's an increase of intestinal permeability. So we are trying to understand what is the role of this septin. And then we did a PCR to look at the gene expression throughout the infection, and we observed that in the 5th day after the infection, we have the highest peak, and then on the 7th day, we have the beginning of the infection, which causes a reduction, and then it increases again at the end of the infection when the animal had developed a hyperparasitemia, where the animal is no longer in a ARDS condition, and the expression of the septin increases again. When we look at the 7th day of infection here, through the predictive model that we had determined, what we observed was that the animals that developed ARDS had a smaller expression of septin 9 in their lungs than the animals that did not develop ARDS. This result corroborated also the expression of septin 9 in the serum of the animals. So there is this modulation profile. In the 5th day here, again, you have a higher expression of this protein in the serum. And in this case, we can separate from the 5th day onwards those who developed ARDS and those who didn't, because we observed the animals until the end of the infection. So saw that on the 5th day, septin is higher in the serum when compared to the animals that did not develop ARDS. This is not the predictive model, but this is an analysis of the cause of death at the end of the infection. And on the 7th day, it reverses back to the same profile that was happening in the lungs. These results, they were from different experiments, and they were reproducible in different experiments. So we're trying to understand these outcomes, and since septin is coded by over 13 genes, and septin 9 has several isoforms. So our hypothesis is that the modulation of these isoforms of septin are different throughout the infection, with different roles as well. So we're now in the phase of identifying the isoforms in the lungs of the infected mice, and here in HUVEC cells, where we're trying to perform in vitro assays. To conclude, we saw that the gene expression and the production of proteins in the serum of septin 9 vary throughout the infection, with peaks at 5 and 17 days post-infection. The septin can be an early biomarker of ARDS and maybe a potential late marker of death, since it increases again at the end of the infection. And up to this point, we have isolated 3 and 4 isoforms of mice and human. And we are searching, this is for the next chapters, we're searching for the differentiation of these isoforms throughout the infection. This is the current group that is working with septin, especially Evental and Camila. And I presented here some results from former students and collaborators of this specific project. Thank you very much for your attention. This is the website of the laboratory. Thank you very much.
Okay, thank you very much for these nice presentations. And we have very, very few time, I see. My watch says 5 minutes for questions. So 10? Good. Okay. So it's open for questions. In Portuguese or English or whatever. Oh, André?
Hello. Thank you, the three of you, for the presentations. I have a question for Maria. I didn't understand in your JAX stat experiments if you use like a lysed erythrocyte control in that conditional media. So my question is, how much of that activation can be driven by erythrocyte endogenous like dumps and not just parasite antigens?
So we always have a control either. So these egress products are kind of difficult to make. So we always have a treated control that we treat red blood cells in the same way on one side. And then I didn't explain it today because I wanted to take over other projects, but we also did the single cell on blood-brain barrier models perfused either with uninfected or infected intact red blood cells. And then we found that there was no changes on the uninfected red blood cells. So we think it's very parasite specific.
But that control for the egress experiment is just media, no lysed erythrocytes, right?
So we've tried both. We have tried lysed erythrocytes and we've tried red blood cells treated in the same way that we used to generate the egress.
Okay, thank you.
Yeah, I also have a question for Maria. So you showed some really exciting new imaging data on endothelial infected red blood cell interactions. And so previously, Michael Lanza and Rick Fairhurst and others have shown that sickle infected red blood cells, they have different knobs and they interact differently and differently activate endothelial cells. Have you looked at these? Would you potentially see some differences?
Sickle cell disease? No, we have not. I think it's very interesting.
Yeah, because you may see some phenotypes that previously people haven't.
Yeah, that would be cool control. Yeah.
Okay, thanks.
Thank you.
I have a question for Maria too. So what's actually in your egress medium? What is in there who does the job? Do you know that? Have you done proteomics with the egress medium?
The question I always get, and it's like something that I've been debating whether to do because there's been many things that have been shown to be disruptive by the parasite. It could be histones, it could be extracellular vesicles, it could be hemozoid. We're also looking at pH now. So there's many things that can be disruptive for the blood-brain barrier. So it could be interesting, but for a PhD student to find that everything is disruptive, maybe it's not so interesting. So that's why we haven't started looking.
Actually, I have another question. Sorry. You have shown that the infected red blood cells really stick firmly to your cells. How do you integrate this with? I've seen papers that show rolling adherence. How is this different? I mean, what I thought I know is that the strongest interaction is with VAR2 CSA and CSA, but your interaction seems much stronger than that.
Yeah, we've tried. We're looking at time course now to see, because we had also this question at Parafrab whether this is what we're seeing is the rolling effects, and then when the parasite rolls, it reorients the knobs. But we've seen that these structures take up to 6 hours to form. So I think it's once the parasite attaches, it remodels the endothelial cells, and this triggers the infected red blood cell remodeling. So I don't think this happens quickly. I think it's a longer process from what we're seeing now in vitro.
More questions?
Hello, I have a question to Maria too. I was really interested about this interaction between the infected red blood cells and the endothelial cells in this cup-like shapes that you form in the endothelial cells. And I was thinking if you measured the type of PFMP1 in your perfusate, and if there's any correlation between different types of PFMP1 and these cup-like forms in the endothelial cells.
Yeah, that's a very question. We have not looked at it yet. We always use clonal parasite lines. This one was expressing PFMP1 that binds to EPCR and ICAM1, because in the past, Anya Jensen and Yvonne Adams also showed that these ones were more actively remodeling the endothelial cell in their organoids. So we haven't looked at other, we have other parasite strains we could look at it. That's a good question.
Thank you.
So no more questions then? Then I would like to thank you all again. And thanks for you listening this interesting session. Vai acontecer agora, não é break? O que vai agora? Oi? Ah, ok.
Obrigada. Vamos continuar então nossa próxima sessão. A Carla e eu
We'll
agradecemos.
continue our next session. Carla and I thank the invitation of the organizing committee, especially Carol and Danielle, to come to this beautiful conference on malaria. And we're going to start with a very important part, with the summaries that were chosen here for oral presentation. I'm going to invite the authors, Igor Mota, Rodrigues de Moura. Please, Igor. Maria Fernanda Alves do Nascimento. Camila de Souza Barbosa.
E Adriele Sacramento de Moraes.
And Adelisa Sacramento de Moraes.
Bom, então a gente começa agora, né, com o
Adriele Sacramento de Moraes. We're going to start now with Igor Mota. Just to remind you, our time here is limited to 6 minutes, so please be brief. Thank you, Igor.
So, hello everyone, good afternoon, my name is Igor, as I was introduced, and I'm going to present a little bit of the work that I did during my PhD. So, to begin with, we all know that malaria is a big problem. This is why we are all here together. And one of the problems of malaria nowadays that I would like to highlight is the spread of resistance to anti-malarial drugs that we have in the clinics nowadays, especially the ACTs. And not all, but almost all of them. And in light of these, we are always looking for new possibilities or targets that we can target and inhibit to kill the parasites. And so, one of these targets that are possible are the proteases within the parasite, in which all the stages of the development of the parasites require at least one protease to be active. For example, here we have the hemoglobin degradation proteases and the egress and invasion proteases. They are essential. And aiming to inhibit these class of proteins, we have the peptidomimetic molecules, which usually show improved drug-like properties and has been used nowadays in the clinics, for example, for HIV, AIDS, actually, as Saquina-V and Nemato-V as for the treatment of COVID. And with that, I would like to show you some of the results that we got. We performed a screen of a few protease inhibitors against the sensitive parasite, the chloroquine-sensitive parasite 3D7, showing quite promising IC50 around 600 nanomolar and no cytotoxicity effect against Hep2 cells. Trying to understand more about this compound that we're going to call it 1153 during the whole presentation. We've performed a panel of resistance in which we test the efficacy of this compound against some resistant strains that we know very well. And interestingly, this compound did not show cross-resistance with the standard antimalarials that confer resistance to the strains that we tested. And more interestingly is that we could see a 10-fold increase in potency when we tested against this compound against the DDT strains that we can see here, compared to 3D7 and even K1. So please keep that information in mind. I'll bring that back again later. So also aiming to understand if this compound could be used in the field, we've tested it against the field circulating strains from Porto Velho, here in Brazil, against both falciparum and Vivax, in which we could see a similar efficacy for falciparum around 100 nanomolar. They're very similar to DDT parasites. But unfortunately, we saw a loss of efficacy when this compound was tested against Plasmodium vivax parasites. Trying to understand a little bit more what was happening with this 10-fold increase in potency for DD2 compared to 3D7 and K1, we were trying to look for the most common differences among these three strains, mainly 3D7, K1, and DD2. And the first difference that caught our attention was the MDR1 copy number increase for DD2 compared to 3D7 and K1 that we can see here, for example. And our hypothesis was maybe this increase, this 10-fold increase in potency could be correlated with this increase in PFMDR1 copy numbers. So to test that, we've used two parasites harboring two copies of MDR1, this gray one, and this yellow one was harboring one copy of the MDR1, in which we saw, when we go from one copy to two copies, we saw an increase in the IC50, and this is the opposite of what we were expecting if the MDR1 copy number increase was correlated to the potency of the compound, to this 10-fold increase. So also, this showed to us that this 10-fold, this increase in MDR1 copy number and increase in IC50 showed to us that maybe there is a target on the cytosol of the parasite, given then that when we increase the copy number, we reduce the concentration of the compound from the cytosol to the DV. Also, aiming to understand more about it, we've performed resistance selections in 3 different backgrounds, and all 3 different backgrounds came back positive out of the resistance selections, showing a similar gene involved in mediating resistance that was falciparum 2A, as we can see here. Falciparum 2A is a hemoglobin degradation protease within the DV, and this information agrees with the fact that under drug exposure, the parasite throws this morphology alteration, in which is very classic for compounds that affect peptide homeostasis for it. Also, we got the 2 mutations that we got after resistance selections and tried to do gene editing using CRISPR-Cas9 in both backgrounds, 3D7 and DD2, in which we observed curiously when we added both the mutations that we got in a 3D7 background conferred a sensitization. The compound became more efficient against the mutant parasites. And on the contrary, when we added, for example, S392R mutation, this is difficult. S392R mutation in the DD2 background, we saw a 10-fold increase in efficacy. This showed to us that falciparum 2A is really mediating resistance to this compound, but also the background in which we have that interferes with the efficacy of this compound. So to finish, I would like to highlight a few conclusion topics, a few bullet points, in which 1153 is a small peptidomimetic molecule with promising antiplasmodial activity and do not show cross-resistance with standard antimalarials, today's strains that we tested. Also, 1153 targets falciparum, but possibly targets another protease within the cytosol of the parasite. Also, falciparum 2A is the main resistance mediator against 1153. MDR1 acts as a low-level resistance mediator, as we saw 2-shift fold by varying MDR1 copy numbers. And also the genetic backgrounds appears to modulate this compound antiplasmodial activity, but the precise basis for that we still don't know and still need to understand properly. And I think understanding what exactly is happening here could help us to find maybe a new pathway, maybe a new target that we could explore for the drug development. And last but not least, I would like to thank all the organizing committees for selecting our work to be presented here, all the financial agencies, and everybody that was involved in this work here in Brazil and doing my PhD scholarship in the Columbia University. So if you want to discuss more about my work, please stop by my poster tomorrow, ending 4:07. I'll be more than happy to discuss more and show you more data that we have so far. Thank you.
Muito obrigada, Igor. Ao final vamos ter
Thank you, Igor. At the end, we are going to have a Q&A session. Now, I'd like to call Maria Fernando do Nascimento from Minas Gerais.
Boa tarde a todos. Meu nome é
Good
Maria
afternoon, everyone. My name is Maria Fernanda.
Boa tarde a todos. Meu nome é Maria Fernanda.
Good afternoon. I'm Maria Fernanda. I'm from the research group on molecular biology and malaria immunology at Fiocruz, Minas, and I'm going to present my work called Expansion of TFH and atypical B-cell subsets associated with long-term immunity to the Plasmodium vivax DBP2 antigen.
A maior parte das cepas circulantes de
Most of the circulating strains of vivax depend on DBP2 for infection, the region 2 ligand that is interacting with the dark surface of the reticulocyte. The region number 2 of DBT is highly polyformic, leading to variant-specific antibody responses. Trying to improve DBP as a vaccine target, John Adams from the South Florida University has developed this vaccine prototype, this a synthetic antigen based on DBP that is directing the immune response for conservated elements called DEC-No2. So neutralizing antibodies against all DBP2 variants, a murine model has shown long-lasting humoral response in individuals exposed to malaria. Knowing the importance of the T follicular importance for the cell B response, our group asked itself, which cellular subsets contribute to long-term, broadly reactive antibody responses against DBP-based antigens? For that, our group conducted this populational basis study. It's been happening for 15 years at this Rio Pardo community. On the top figure in green, we see that the sample collection were made in low and high transmission moments. And in the lower figure, we can see that in the end of our cohort, based on DEC-No2 and DBP elements, we identified responsive and non-responsive elements. What I'm going to show to you is a part of our study at the end of our cohort, when the individuals had no detectable malaria, no parasitemia, they were negative in PCR, and DBMC were stimulated in culture for 6 days and analyzed with flow cytometry. And the first result that I'm going to show to you, you may see in red the resistant responder according to the two antigens, reminding you that the follicular T cell help produce antibodies and T cells. We evaluated several subtypes of T cells with subtype TFH1 and and 1 like and 17. And we had the increase of persistent compared to the other tested antigen. And the Th1 response was reported in literature as important for Vivex malaria. And we've seen the subtype Th2 and Th17 due to the fact of the individuals being PCR negative in a persistent way. We also evaluated several B cell populations, but we called the attention for atypical B cells. These cells have been reported in literature, and this marker, the first that is positive, it was enhanced in the persistent respondent in red compared to the other antigens. And we observed here in this unprecedented study, this CXCR3 positive for individuals in red who were persistent responder for BP antigens. And the importance of the atypical B cells was reported in recent literature with symptomatic malaria. We also had no supervised analysis of the samples with the collaboration of Dr. Gregorio from Fiocruz, Minas Gerais. And we may see in red that the CXCR5 positive was, and the cells were not the classic follicular cell, but other T types that should be more investigated. We also evaluated several clusters of B cells, and I'd like to draw the attention in yellow, the same markers that was CXCR5 positive expanded in this cluster of B cells reported for the first time in the Vivex malaria. We also had this correlation analysis between the different clusters, and we could see that the persistent respondents had a smaller response from the cells, showing a modulation of the immune response. Our work is showing that the immune response for malaria is highly complex, and what we saw in our study was the increase of subtypes Th1, Th2, Th17 in the resistant one, and CXCR5 B cell positive and atypical Tibet cells and CXCR5 positive. And all this set was contributing somehow for the generation and maintenance of antibody response in a long-term and high affinity. I'd like to acknowledge all of our collaborators, our funding agencies, and the Congress for the opportunity to present this work to you. Thank you so much.
Acho que agora sim. Então, agora a nossa próxima é a
Now,
Camila de
the
Souza.
next one is going to be Camila de Souza from Unicesp, Escola Paulista de Medicina, Medicine School, from Sao Paulo. Good afternoon, everyone. I'd like to thank the organizing committee for this opportunity to present my work that is called the Coconate as an Anti-Plasmodial Candidate Against Resistance to Artemisinin-Based Combination Therapies. Malaria is one of the greatest problems we have. We have the resistant parasites to available treatment, and in this map we show the failure of treatment in artemether lumefantrine treatment between 2015 and 2026, and we see an increase of studies reporting an increase of up to 5% in treatment failure, especially in Africa. Concerning Brazil, I brought data from our lab where we had in vitro and ex vivo assays to determine the successibility of isolated from Roraima in face of antimalarial described in this chart. And we can notice that the falciparum is in blue. We have some isolated where we saw a lower susceptibility to drug, and Vivax are in green, and in all cases, we had more susceptibility to the antimalarial tested. If you want more information, you may see these two posters, and the students are going to talk more about that. And this is reinforcing the development of new drugs, and my work was to evaluate the coconut to determine the uh, parasite profile. And the coconut has this chemical structure here. It's a compound that comes from repositioning, and we have several reports in the literature showing its anti-plasmodial uh, activity in the face of different parasites, and this compound it will present these these activities of TCP1, 4, and 5, but not everything is positive. There's this great disadvantage for this compound, and we have a low solubility in water for several organic compounds, but there are two strategies to revert that: structural strategies. And here is a piece of data showing that the nanoliposome of the coconut has shown an inhibitory promising effect, and also reduce the parasites with a dose of 10 milligrams per kilogram, and also they reported a long-lasting effect in Sprague Dawley rats. So here I'm going to show you the results we obtained in lab. And at first, we wanted to determine if this compound has a fast or slow action. First, we had this the speed of action assay. So you may see IC50 defined after exposure in different times. We had the parasite being exposed in different times, and the coconut has presented a profile that was similar to a tovacone. 48 hours for this maximum effect, and this is showing us that this is a slow compound. And figure 5, we have the preliminary results of the stage specificity assay, and this compound has this lower inhibitor that is corroborating with the previous data. The next step was to have the in vitro evolution of the parasite to see if they would be resistant to the compound. We were able to get the parasites, and as described in literature, this way of action of this compound was via cytochrome B. We had the gene sequencing, and we had identified the mutation V259L, and this mutation was described in literature. And in figure number 6, we have the growth curves of tovacone at the top and the coconut at the bottom, with the two strains, one generated in lab and the GT2 that was described in the article with the same mutation. And we are able to see a shift in the C50 values, but the shifts are slightly different. Last, in this figure number 7, we are showing the place of mutation at cytochrome B, the site key 0 of this gene.
So as a conclusion, this compound is a promising antiplasmodic candidate. It has activities TCP1, TCP4, and TCP5, which makes it a promising candidate. And an interesting fact is that this mutation V259L in this paper here was demonstrated that it is not transmitted to the mosquito. So if there is a transmission, the number of oocytes that are produced is way smaller, which guarantees a certain security in the use of this compound. As perspectives, we are still going to do a whole genome sequencing to see if this compound is going to generate new mutations, and a transcriptomic analysis to determine how this resistance mechanism happened. I would like to thank my supervisor, Ana Caroline Campos Aguiar, the collaborators, Professor Diogo Moreira, and PhD Caroline Conceição Souza from Fiocruz, Bahia. Thank you for the collaboration, and my lab mates. the institutions and the fostering organizations. Thank you. Thank you, Camila. Now we have Adriele Sacramento Moraes from Fiocruz, Bahia.
Boa tarde a todas e a todos.
Good afternoon, everyone. My name is Adriele Sacramento. I'm from Fiocruz, Bahia, and I'm going to present the work that I developed during my master's called Antiplasmodial Evaluation of New Artemisinin Derivatives Against Plasmodium falciparum strains partially resistant to ARTs. The colleagues discussed a little bit about the scenario. So speaking about the history, in 2006, we identified in the Southeast Asia an important clinical milestone of resistance to artemisinin, which is this delayed parasite clearance in ACT-treated patients. This milestone is also known as partial resistance to artemisinin, and concomitant to that, there was a development of the laboratory strain. And from the sequencing of the genome, we have identified one of the main genetic markers for ART resistance, which is CALCH13. Today, we know that this profile of partial resistance is not restricted to the region of Southeast Asia. We have other hotspots in the African continent. In this perspective, my work seeks to understand a little bit more how novel peroxide derivatives work through ALH325. Have here two conjugated artemisinins forming this dimer that has a long-lasting profile, one of the Achilles heels of artemisinin. It was developed in partnership with Dr. Svetlana from the University of Nuremberg. We did an in vivo, in vitro, and stability characterization of this compound. It is identified as compound 3, and we tested its potency in vitro for strains that are sensitive to chloroquine and resistant, chloroquine-resistant strains like DD2 and K1. So it proved to be below 5 nanomolar. In this profile, we created an in vivo correlation where the compound could achieve 100% cure in mice that was infected with Plasmodium burgii. To understand the stability of these dimers of this pharmaceutical, LH325, we did a stability study where we compared in 24 hours the stability of this compound. To be easier to explain, we have RDHA, which is the base compound. In 24 hours, we have the degradation. And torvacone, which we know it is a long-duration pharmaceutical, which continues in 24 hours with the equivalent 500 nanomolar. Compound 3 is in the middle exactly, so it still has a decrease in its stability because it derives from artemisinin, but it has this profile of being a peroxide with a slower duration. We would like to understand how it works in the scenario of our ART resistance. I went to the laboratory of Professor Benoit Vincal and Lucie Paloc in France to do assays in the resistant strain where we did the studies of survival of the ring, and we identified that the compound was able to kill these resistant parasites, and the survival rate was 2.5% in comparison to the hydroartemisinin that had a survival rate of 8.3%. In this preliminary study of in the sensitive strain, we saw that the traces is that there's no cross resistance at the time. And since this compound is killing the resistant parasite, but does it also induce the quiescence profile that artemisinin does? So we did a survey where we treated these resistant parasites for 48 hours. And in the middle of these 48 hours, we did 2 sorbitol administrations. So if the parasite entered quiescence, the treatment with sorbitol is not going to affect the recrudescence of this parasite. What we saw is that both compounds of artemisinin, DHA, and ours have a very similar phenotype in microscopy, where these rings become pycnotic in comparison with dorvacone that has long duration. The cycle continues developing. So what we noticed was, as expected, artemisinin induces, LH325 induces quiescence with long-acting profiles. Since it is derived from artemisinin, what we noticed was that the recrudescence of the parasites treated with LH325 is always later because of this long duration effect, probably. Our next steps then are to understand what distinguishes the activity of this compound in comparison with DHA. They are molecularly similar, but this dimerization causes a consequence that is killing these resistant parasites in a different form. So in order to understand this, we're going to do a transcriptome analysis of the parasites treated and unveil a little bit more of the mechanism of action and how these long-duration parasites work. Thank you very much to the Commission for the opportunity to show my work and all of the collaborators.
Bom, agora a gente abre para
Now we're going to open for questions. We have 6 minutes.
Eu vou fazer uma para.
I'm going to ask a question. There's a question here from Silvia. Silvina, please. I'd like to ask a question to Maria Fernanda. I thought your data was very interesting, but I was curious. When you do the phenotyping of the TSHs, you leave them for a while in a culture. How do you add something? How do you do this culture? We stimulated for 6 days with the antigen of DBP. Only the antigen. You don't add cytokines. No, because since we're using PBMC in a total form, we believe that there could be monocytes and they could form a microenvironment that could help in the secretion of cytokines. That is why we don't use them. Thank you very much. Anybody else would like to ask a question? No? I will ask a question to you, Arielle. When you tested your compound, because it is an artemisinin-derived compound, did you test other doses or you fixed the dose to avoid recrudescence? Because you might administer a very high dose, it eliminates, and then it induces again. So did you test other doses? I went to Professor Francois' laboratory for 3 months, so we could not do this final testing with other concentrations. In this experiment, we used 700 nanomolar to be equivalent to our assay, but we intend to try to reduce this dose and see how it will work. What would be the response? Thank you. I have a question to Maria Fernanda. Congratulations for your work. Could you see if there's any difference in patients that had several episodes of malaria, more than 5 or 6, or patients that had 1 episode of malaria? Since we did the study at the end of our cohort, the individuals did not have parasitemia anymore. There has been a long time that they did not have parasitemia. In the beginning of the study, some of the individuals did, but we did not see a difference because at the moment that we did the study, none of them had parasitemia anymore. Even through PCR, they were negative.
Mais alguém? Alguma coisa?
Anybody else? No? So I think we have wrapped up the session. We would like to congratulate the candidates that were selected. It's an honor to be here, so good luck and thank you very much. Now we're going to move forward to a coffee break. We're going to have a poster session, which happens together with the coffee break. This is another opportunity for us to discuss with the presenters. Thank you.